Hello everyone, and welcome back to AP Bio with AP Playground. Today we're covering topic 6.8, Biotechnology. Today we need to explain the use of genetic engineering techniques in analyzing or manipulating DNA, and we'll be looking at 4 examples of the following, including gel electrophoresis, polymerase chain reaction, bacterial transformation, and DNA sequencing. And suggested skill is argumentation 6D. Explain the relationship between experimental results and larger biological concepts, processes, and/or theories. Biotechnology is the process of manipulating organisms for the purpose of making useful products. And we'll be looking specifically today at manipulating genomes in order to understand or possibly get specific results from these genomes. So first up is DNA sequencing, and this is a fairly recent discovery, but we've been able to sequence the entire human genome done by the Human Genome Project, and it's used to determine the complete nucleotide sequence of a DNA molecule using base pairing. And it allows for comparison of DNA between species. And what DNA sequencing has taught us was how similar we are to many other species. For instance, humans and potatoes share 50% of their genomes. Humans and pigs share 98% of their genomes. And this is all evidence for common ancestry because of how universal and how much of the genome is shared between these fairly seemingly unrelated organisms. So gel electrophoresis is a lab technique used to separate DNA, where DNA is placed in a gel matrix that allows small molecules to move through it. An electric current is applied to the field, and DNA is negatively charged due to the phosphate groups in nucleotides. This means that the DNA fragments will be migrating, and they'll be attracted to the positive end of the field that the current is applied through. And smaller fragments are going to move the fastest. And this results in banding patterns, and the differences within these banding patterns allows for diagnosis of disease, to determine who the parents of a child are, and also for crime scenes. So if we look here, we have DNA collected from a crime scene, and we also have DNA of a few suspects. You can see that suspect 3's banding patterns match up with the crime scene's banding patterns, the DNA found at the crime scene. So gel electrophoresis is extremely helpful for this. And a child's DNA is going to be a combination of maternal and paternal offspring, so maternal and paternal DNA. So if you look at different candidates for one of the parents, you're able to tell by comparing it to the banding patterns of the child's DNA who could be the parent of the child. Next up is PCR, or polymerase chain reaction, which allows amplification of a piece of DNA without using cells. So without going through cell division, we can amplify a piece of DNA and do some insane replication. And so first of all, you have to quickly heat the DNA in order to denature and separate the DNA strands. Then there's some cooling done so primers can attach, and then DNA polymerase is introduced and adds nucleotides to the 3' end of the primer. And so we get a bunch of identical DNA molecules. So if you look here, here's our DNA with the sequence of interest. It's heated up and denatured, then primer attaches, does base pairing, and now we have, after the first cycle, we have replicated piece of DNA. Then this same thing can be repeated. We have heating, we have cooling, and then we have DNA polymerase adding nucleotides. So after the second cycle, we have 4 copies of this DNA sequence of interest, and so forth and so forth. After 20 to 30 cycles, we can have millions of copies by performing PCR. Last up is recombinant DNA and gene cloning. Recombinant DNA is artificially made DNA using DNA from different sources, such as introducing human genes into bacteria. And this is done through the use of plasmid, which is an extra chromosomal small circular piece of DNA. And plasmids can be entered into bacteria, then they can be taken up into the overall bacterial genome, and then these genes can be expressed. And now we have recombinant DNA that's mixing both bacterial and human DNA. Now, genes can also be cloned with the use of plasmids and bacteria, and this is the process by which scientists can produce multiple copies of specific segments of DNA. If the specific segment of DNA is turned into a plasmid and entered into a bacteria, bacteria are going to reproduce a lot faster than human cells. So all of a sudden, you can have a lot more copies of this specific gene. This is gene cloning. and cutting apart to get this specific segment of DNA happens through restriction enzymes such as CRISPR-Cas9. It cuts DNA at a specific location, resulting in restriction fragments. So you can see here the restriction enzyme is cutting right over here, and that's going to result in some sticky ends. These are where there are nucleotides that are not base paired. So they're kind of hanging out the end, they're a little sticky. And they can bond to complementary strands using DNA ligase. So DNA ligase comes in and finds sticky ends that it can bind together and sort of acts as the glue to help these sticky ends bind and create complementary strands. So this way, in this way, restriction enzymes are able to cut out specific sequences of DNA, which can then be turned into plasmids and cloned by bacteria. So today, in the last lesson of Unit 6, we discussed methods and benefits of dealing with DNA using biotechnology, including DNA sequencing, PCR, gel electrophoresis, and gene cloning. We'll see you in Unit 7 to talk about natural selection.